Okay. Good afternoon, ladies and gentlemen. My name is Roman Ovchinnikov and I'm happy to participate in this conference. And I'll briefly touch on fungal pathogens in wild Siberian sables detected by culturomic and next-gen metagenomic sequencing. Did you know that in wild nature sables live only in two countries of the world? It's Russia and Japan. But it's well known that sables are valuable fur-bearing animals and they are the object of the fur trade. Unfortunately, however, we know very little about infectious disease in these cute wild animals. So me and my colleagues from Tomsk State University in Siberia region were happy to catch the chance and to perform this research project. Next slide, please. So let me give you some background. Mass skin lesions in wild Siberian sables have been reported since 18th century. In affected animals, hair stick together and falls out, so the quality of fur decreased dramatically. The prevalence of the disease is high, and according to recent data, more than 50% of procured sable skins are damaged due to this infection. Next slide, please. An important question is what is the main cause of the pathology? Despite long history of the disease, true etiology is still unknown. First mycological study was performed more than 60 years, and this study revealed high occurrence of acrimonium fungi in majority of affected sables. So acrimonium was judged as a causative agent of the disease, but no confirmative studies were performed in subsequent 60 years. Next, please. So based on this, the aim of our study was to detect potentially pathogenic fungi in skin lesions of affected sables and to confirm the fungal etiology of the disease. To conduct the study, 15 hair samples were collected from affected skins in Tomsk region in Siberia and delivered to our laboratory in Moscow to perform the mycological examination. Next, please. Here we can see the hair samples from affected sables. We can see that hairs are glued in bands with crusts, and on microscopy abundant pus-like debris is observed. So these features indicate the presence of intensive inflammation in affected sites of damaged skin. Next slide, please. Okay, the mycological examination was performed using both conventional and NGS metagenomic approaches. Conventional approach included cultural study of samples on saburo and DTM type media with subsequent identification of isolated cultures. And another portion of samples was subjected for metagenomic NGS sequencing targeting ITS region on Illumina MiSeq instrument, and this metagenomic study was performed in Commercial Genomic Center. Next, please. So here we can see the interactive diagram of metagenomic NGS analysis. And first of all, it should be noted that 3 samples were subjected to NGS and only one of them yielded a sufficient quantity of fungal DNA to perform NGS analysis. In fact, we were able to get the results for only one sample. And here we can see that totally 249 operational taxonomic units were detected in the sample by NGS. Naturally, we were very impressed with such a variety of fungi in single hair sample, but it will take a lot of time to review all detected species, although it's very interesting, of course. Well, since today we are talking about Onygenalean fungi, it's important for us to point that 9% of detected fungi belonged to order Onygenales, and you can see them on the left side of the diagram. Next, please. So let's see in detail the results concerning Onygenales. we can see that 81% of Onygenales were occupied by Incerticedes families of uncertain taxonomic attribution, and we will consider them a little bit later. So 18% in Onygenales were represented by Arthrodermataceae family, and 0.5% were taken by Agelomycetaceae family. So if we take a look at Arthrodermataceae, we will see that it was presented by Arthroderma and Trichophyton genus, and the only species of Arthroderma was Arthroderma insingulare, and Trichophyton genus was presented by Trichophyton anihocola and identified species of Trichophyton. It should be noted that this Trichophyton species was closely related to Arthroderma cuniculi, as it was related by blast verification, and you can see it on this slide. So next slide, please. Well, in Incerticedes families, the genus Chrysosporium was dominating, and it included species Chrysosporium carmichaeli, Chrysosporium undulatum, and one more unidentified Chrysosporium species. Also, the genus Malbranchia was presented in this section, and it consisted of one species, Malbranchia cinnamomea. And finally, Agelomycetaceae family was presented by genus Emontia, which consisted of Emontia helica only, the possible agent of Aedeospiromycosis in animals. So we can see a significant variety of potentially pathogenic Onygenalean fungi were detected by targeting metagenomic sequencing in a single hair sample. Next slide, please. Well, now let's take a look on brief results of mycological culture. Overall, 31 fungal cultures were isolated from 15 samples, and wherein only 3 cultures belonged to order Onygenales. Next, please. Well, 2 of isolated Onygenalean fungi revealed the closest homology to Arthroderma cuniculi, and you can see this culture on this slide. It's important to know that primary cultures of Onygenalean fungi were isolated on DTM media, as you can see on left picture, but not on Saburo media. At the same time, on Saburo a variety of mold fungi were grown, including numerous colonies of Acrimonium species. Going back to the story I told you earlier, the primary studies identified Acrimonium fungi as causative agent of dermatitis in sables 60 years ago. But we can see that those researchers didn't use selective media such as DTM media because it was just not available at the time, so they really were not able to isolate dermatophytic fungi, which are the most likely causative agent of skin disease, while Acrimonium and other numerous non-dermatophytic molds are probably just skin contaminants. Next, please. Well, one isolated strain of Arthroderma cuniculi was subjected to whole genome sequencing on Illumina MiSeq platform. So here you can see the draft results of the de novo genome assembling. And for now, we think that this strain represents the new species of Arthroderma. So Arthroderma cuniculi is just our preliminary identification. And in the nearest future, we are planning to analyze this genome in detail and to clarify the taxonomic attribution of the species in collaboration with our Czech colleague Vít Hubka. Next, please. So here we can see that one more Onygenalean species isolated from sables was Chrysosporium carmichaeli, which was the most prevalent Onygenalean species on NGS study. And it was isolated only from one sample on DTM selective media. You can see it on the left slide, and the colony, white colony, is surrounded by gray mold fungi. So just to recap, we obtained 3 cultures of Onygenalean fungi from 15 samples, and each of them was isolated from individual skin sable sample from the sable. So, next slide please. Is there any correlation between metagenomic and cultural results? Well, on the one hand, we were able to isolate the cultures of 2 Onygenalean species, which were detected by also by metagenomic sequencing. But on the other hand, 6 other Onygenalean species detected by metagenomic were not isolated in culture. And we should remember, of course, that only 1 sample was analyzed by metagenomic, so our findings should be confirmed in future studies, of course. And next slide, please. Well, in conclusion, personally, I feel that the most important result of NGS sequencing is the fact that in a single affected animal we can find not the only one dermatophytic pathogen, but plentiful bunch, plentiful association of keratinophilic fungi. But which species is primary pathogen, which one is secondary pathogen, and which one is just geophilic fungus? All these questions are very interesting, and we are going to explore them in our future studies. Anyway, I believe that targeting metagenomic sequencing will be optimized for using with clinical samples, especially hair samples, skin samples, and these analyses will become more cheaper and more accessible for routine diagnostic. And of course, this approach opens up great prospects in diagnosing of fungal infections, both in animals and in humans. Final slide, please. So, thank you very much for your attention, and you are very welcome to contact us for any question. Thank you very much.